Questions. We'll move on to the next speaker, and let me try and get this. Okay. So how do I get rid of her? I can just slow this. Okay. Second speaker, I think, is Dr. Chang. So our next speaker is Dr. Shyang Chang, who will give us an unexpected view, unexpected perspective on the mechanism of action of acupuncture.
Thank you, Mr. Chairman, ladies and gentlemen. It will be a great pleasure for me to be here to present what I've been doing during the past 20 years in 20 minutes. And since I have only 20 minutes, well, first the topic I'm going to talk about today is challenging the basic tenets in neuroscience, lessons from Chinese acupuncture. And I will put down the 5 main things in the very beginning. Number 1, human body is actually a complex dynamic system. Number 2, modern cellular or molecular medicine concentrates more on the structure rather than the function. Number 3, however, function is more essential than the structure in medicine because the structure, when structure changes, the function can adapt. It has always been said that harmony, health is harmony, disease is discord. Even though the harmony might be a minor key, which is a comment made by Edwin. And number 4, acupuncture or traditional Chinese medicine, TCM, treats disease from the dynamical or functional point of view in the very beginning. So number 5, the idea of TCM, if we use it in modern notions, and I believe it can promote world health and reduce the healthcare cost. In order to validate my claim, I'm going to divide the talk today into 4 parts. Number 1, introduction. Number 2, mathematical foundation. And then experimental results. And number 4, brief conclusion. So there are actually 2 basic tenets in neuroscience. The first one is the neural doctrine. And as we know from this morning's keynote speaker, that Gorgi actually has different views with Cahal, especially in the dynamic polarization part. And another one is the mutual inhibition. This is more on function. Actually, these 2 are intertwined, the mutual inhibition. And I have also put down Hodgkin and Huxley's leaky cable nerve fibers, leaky theory of the nerve fibers together. And we will try to re-evaluate these ideas in today's talk. So let's start with the inhibition. In 1846, Weber's 3 brothers actually used tentative excitation through the nostril and the 4th vertebra of the frog and caused the standstill of the heart. Because the nostril is very close to vagus nerve, so they believe that vagus nerve has the power of inhibition to the heart. And this idea has been passed on to Valentin during micturition. And he believed that external urethral sphincter will voluntarily relax while the detrusor will contract. And Sherrington believes not only the heart muscle, the smooth muscle, but also the skeletal muscles will have this reciprocal inhibition. And Penfield went to Sherrington's lab and learned a lot of the techniques. And it turns out it's the Renshaw cell that has been used by Eccles to explain his professors, his mentors' idea of mutual inhibition. He believed, and his followers believed, that the presynaptic and postsynaptic synapses will play a role. And these ideas permeate in neuroscience from the respiratory rhythmogenesis and even to higher functions of the brain right now. And this is the Renshaw cell in the spinal cord. And in 2007, Journal of Physiology, Dr. Alvarez and his colleagues actually had a paper, The Continuum Case for the Renshaw cell. He said, despite the detailed information generated in the 50s and 60s, there remained controversy over the existence of Renshaw cell and their role in the recurrent inhibition. And he furthermore pointed out, it's been difficult to directly test these and related hypotheses because it's very hard to antagonize, delete, or knock out ventral cells to monitor their behavior in freely moving, non-anesthetized animals or human beings. And if you look at his paper, no matter intracellular or extracellular or paracellular recordings, you will see nothing but rhythms, rhythms, and rhythms. So let's come to the second part. The basic idea of TCM, traditional Chinese medicine, and acupuncture are actually rhythmic in the very beginning, because in a agrarian society, night follows the day, and the tides ebb and flow, seasons change, visible and invisible rhythms underline the world around us. So man's place is only a humble part of the periodic time cycle. And physiological rhythms usually interact not just with one another, and probably with the outside environment. And we call disease if the normal rhythms break down or new abnormal rhythms occur. And this has actually been the essential idea of dynamic disease proposed in the 1970s. And so since rhythms are important, so the mathematical foundation I have proposed is to characterize rhythms in physiological functions. They have to be collective amplitudes and frequencies, because rhythms have to be characterized by the amplitudes and the frequencies. The only thing that's different from a single neuron or single nerve fiber is that because we have now a collection of fibers and neurons, etc. So we have proposed the following tools. In order to capture the amplitudes, we use the generalization of Brownian motion. It's called the fractional Brownian motion, and its increment, the fractional Gaussian noise. In order to capture its frequencies, we use the spectral measure. And we don't have time to go into detail, so here's the Brownian motion, here's the fractional Brownian motion, and I'm going to explain the meaning, physiological meaning of fractional dimensioning at the end. It's actually this h that we're going to extract for, because if h is greater than 0.5, the interpretation is that the amplitudes are actually positively correlated, it's persistent. If h is less than 0.5, the amplitude is actually anti-persistent or negatively correlated, and the frequency will be derived from this part, the spectral measure, and actually there are 3 parts from the spectral measure. If you know a little mathematics, you will know it. There are 3 parts, the continuous part, the absolute continuous part, and the singular part. Singular part will be ones that we're interested in. Now, let's look at the experimental results. We're going to present 3 experimental results, but the emphasis will be put on the acupuncture. First is the maturation and urine storage of the lower urinary tract. This is an animal study with star rats. And then elbow flexion and forearm pronation in humans, and effects of acupuncture. And this is the bladder, and this is EUS, external urethral sphincter. They're innervated by sympathetic and parasympathetic to the detrusor, and the EUS is innervated by pudendal nerve. And it's the general belief that when you micturate, the EUS has to be relaxed and the detrusor will contract. And during urine storage, that the EUS has to contract and be in synergy with the sympathetic. But we have seen actually the opposite. If you look at Campbell's urology book, even the page 6th and 7th edition, this is what they have for, this is what they have for the, EU, EMG, and actually it's bursting, but the general belief was it has to be quiescent. So they actually cut it out and replaced by a straight line. But this is not the case. If you look at the animal studies, you will see that this is the bursting during micturition, and this is the cystometrogram, this is the electromyogram of the EMG, and this is the SCIE rats. Even though you still have bursting, but it's not efficient. So whenever you see the color code from green to blue, it means that the amplitudes or the collective strength has been emphasized or has been strengthened. So whenever you see